RDP in this video. So basically, reference-based alignment, pre-taxonomic classification. So RDP classifier, firstly, naive Bayesian approach. Basically, what is the core idea here is that instead of aligning a query sequence to references, slow, computationally heavy, the classifier uses k-mer frequencies. It treats the problem as what is the probability this reads. This read belongs to genus G given its set of k-mers. How it works in mathematics. So build the naive Bayesian models for each genus, training on all sequences in reference, for example, RDP database. For each k-mer, word of length k, typically k is equal to 8, compute m of wi, which is the number of times the word wi appears in genus G's sequences. Compute conditional probability p wi given G. For a query sequence S, break into k-mers. For each genus G, compute this. For each genus G, P of S given G is actually directly proportional to the product of the conditional probability of Wi given G, and there this is the product from 1 to n. Then compare across genera, assign to genus with the highest probability. Then bootstrap for confidence. Instead of using all k-mers, take random subsets of k-mers, bootstrap resampling, repeat 100 plus times, count how often each taxonomic node, that is domain, phylum, genus, is recovered, then report the confidence scores for each level. Now, strengths and weaknesses. So, it is fast scalable for larger data sets. Probabilistic provides confidence estimates and widely used in query pipelines. But weakness is that it performs best at genus level, less reliable at the species level. 16s lacks resolution. Then, dependent on completeness and accuracy of reference database, we have syntax k-mer overlap based. Right. Core idea is similar to RDP but not Bayesian. It uses k-mer overlap between query and reference database. It assigns taxonomy based on the highest similarity score. Uses bootstrap resampling for confidence like we do in RDP. Key features are works at species level resolution also, claims higher granularity than RDP. Simpler model, no prior probabilities required. For example, query has 100 k-mers, 90 match best to Bacillus subtilis sequences. Bootstrap across subsets gives 95% confidence. Then we have Spingo. So the core idea again, it is like syntax k-mer overlap algorithm designed specifically for species level classification. Each sequence is matched to a reference by maximizing the overlap score in Spingo. Bootstrap computes the confidence scores. So key features, optimized for 16s and ITS species level classification and faster than RDP, and works well in low complexity environments, that is few dominant species. Now moving on, comparing the three. So we have the feature, the RDP classifier, syntax, and Spingo. Three classifiers are going to be compared. So model in RDP is basically naive Bayesian. In our syntax is k-mer overlap, and Spingo is also k-mer overlap. Target database for RDP is RDP 16s. Then syntax is flexible, 16s ITS. Spingo is 16s and ITS. Granularity, genus level reliable. Syntax is species level capable, and Spingo is also species level capable. So no difference between the two till the granularity, right? And all of these points are same for the two. Now confidence, bootstrap resampling for all three, same. Strengths, well established confidence at each node, widely used. Syntax, simple, fast, claims better species resolution. Spingo is species level optimized and is lightweight. Weakness for RDP is limited species resolution, older training sets. Syntax dependent on reference may overclassify, and Spingo accuracy drops in complex communities. Now, practical example in microbiome analysis, 16s gut microbiome study, Illumina MySec 2 cross 250, reads denoised into ASVs via data 2. Classification pipeline uses RDP classifier genus level for broad taxonomic assignment. Syntax for Spingo species level for refined species call if the database coverage is strong. Final output is abundance table with taxonomy plus the confidence scores. So in summary, RDP classifier uses naive Bayesian algorithm, strong at the genus level, provides confidence bootstraps. Syntax and Spingo are alignment free, k-mer overlap models, better at the species level depending on database quality. All use the bootstrap resampling to provide statistical confidence in taxonomy assignment. Now moving on, we'll be talking about Spingo and the reference-based taxonomies, right? So example of Spingo, k-mer overlap. Spingo algorithm is species level identification of microbial sequences using the k-mer overlap. Score computation, let query q is equal to q and reference is equal to r. Define qk, set of all k-mers in a query, and rk as the set of all k-mers in reference. All k-mers in query q and all k-mers in reference r. Overlap score would be qk q subscript. This is the subscript. Okay, so that would be qk intersection rk upon qk and the number. Since we are considering the score, we have basically the absolute of this. So this basically measures how many query k-mers are explained by the reference sequence. And we can have an example here. Query 50 base pairs, k is equal to 8, qk is 43 k-mers, and reference has 40 of those k-mers. So score becomes, since we are taking the intersection, so 40 upon 43.93, very high, likely species level match. Strength focuses on species level resolution, unlike RDP, which is genus focused. Weakness is it needs high quality reference database, else may overcall species. Alignment based reference classification, unlike RDP, Spingo, or syntax, which are alignment free, we have alignment based methods also, which directly align reads to reference sequences. Typical tool we use is BLAST nucleotide BLAST. Workflow is query aligned against reference, Silva, green genes, RefSec. Best hit, highest percentage identity, lowest e value chosen, and then assign taxonomy accordingly. Problem is that if multiple hits have the same percentage identity, how will we assign? So the solution is that we use lowest common ancestor rule, LCA rule, assign the query to the lowest taxonomic rank that is common across all the best hits. For example, query matches equally well to E. coli and Shigella flexneri, then LCA would be the family Enterobacteria, say. Strength, high accuracy if reference contains true homologue, and weakness is computationally expensive, ambiguous when multiple hits exist. Then operational taxonomic units, OTUs. Definition is an operational definition grouping sequences by similarity, often used as proxy for species in 16S studies. Standard threshold 97% sequence identity. Sequences greater than 97% identical and have the same OTU, operational taxonomic unit. Example, two 16S sequences share 98% identity, one OTU, if only 95% separate OTUs. Different OTU picking modes. De novo OTU picking, cluster reads purely by similarity, no reference needed. Useful for novel environments, but produce study-specific OTUs. Then we have closed reference OTU picking, where only cluster reads that match a reference database, for example, green genes. Sequencing without a reference, discarded. And reproducible across studies, but may lose novel taxa. Open reference OTU picking, hybrid, first match to reference OTUs, then cluster remaining sequences de novo. So we get best of both worlds. How do we form clusters? So complete linkage, that is the furthest neighbor. All sequences in the cluster must be less than equal to the threshold from every other sequence. Complete linkage, furthest neighbor. Single linkage, nearest neighbor. Each sequence must be less than equal to threshold from at least one sequence in the cluster. Then average linkage, average pairwise distance between sequences must be less than threshold. So these are the three formats, the three forms of clusters: complete linkage, single linkage, and average linkage. Now the problem is these require all pairwise distances, so it is not scalable, and it is sensitive to new data. Adding sequences may reorganize the clusters. So we use a greedy centroid based clustering algorithm. So practical alternative is sort the sequences by abundance or length. Take the first sequence, cluster centroid. For each new sequence, if similar enough to an existing centroid, just join the cluster. Else we create a new cluster centroid. Representative sequences are centroids and widely implemented in CD8, UCL, UCL, UClust, UCluster, and USearch. Example: 1 million reads centroid based clustering can group into 10,000 OTUs efficiently. Now pipeline for amplicon data analysis. So these integrate all the steps from raw reads to OTUs, ASVs to taxonomy tables. So we have QIIME2, which is modern modular framework, supports both OTUs, research, and ASVs, data2 and deblur. Taxonomy assignment, RDP classifier, syntax, Silva, train, naive bias. Then outputs, feature tables, taxonomic bar plots, diversity analysis. Then we have Mother, older but still widely used, uses clustering, quality filtering, chimera detection, RDP like classifiers for taxonomy. Then we use vSearch, open source alternative to usearch, implements greedy centroid OTU clustering, can also do chimera detection, UKIME, deep replication, and fast searching. Now pipeline steps overview. So first we have the stage of quality processing. In QIIME2 we use cut adapt, demultiplexing, demux, data2 deblur, right, for ASVs. Then in Mother we use trim.sequences and vSearch fastQ filter. In chimera removal, data2 vSearch UKIME for QIIME2. Mother pintail bellerophone chimera.share. vSearch we use UKIME. And let us go to the important part, that is the output. So in output, QIIME2 gives interactive visualizations, feature tables. Mother gives OTU tables, and vSearch gives OTU tables as well. So summaries: Pingo uses k-mer overlap for species level assignment. Alignment-based methods, BLAST and LCA, are accurate but slow. OTUs group sequences by similarity, now largely replaced by ASVs, data2 and deblur. Pipelines, QIIME2, Mother, vSearch automate full workflow from raw reads to taxonomy.